Thank you. We now have about 10 minutes for questions, and if you could move to the microphone for the purpose of taping, and please identify yourself with your questions.
Jan Patterson, San Antonio. So, Dr. Girard, as an infectious disease physician, we are increasingly seeing multi-drug resistant pathogens that are resistant to all our commonly used antibiotics. And as we've seen more of that, the production of new anti-infectives, particularly for gram-negatives, has come to a virtual standstill. So I was wondering if your operation is going to be looking at those kinds of pharmaceuticals, and also I'm interested in your thoughts about what strategies would stimulate interest in those anti-infectives.
Thank you for the question. And of course, it's one of the key questions that's being faced throughout the country and throughout the world, and that is the lack of effective therapeutics against multi-drug resistance, but also the lack of a pipeline to approach those resistant organisms. I'll say simply, what I talked about now is not dealing with that. It's dealing with biological therapeutics and vaccines. That being said, there are interdisciplinary programs and interinstitutional programs out of one of our centers from Jim Saccatini, who is at least looking at what's probably the world's largest natural product library derived from unusual microbes and microbial products, looking at both unconventional pathogens as well as standard pathogens. And he's one of the people who has 70 people and spans from discovery, looking at whole cell hits through co-crystallography, through medicinal chemistry. But perhaps someone else in the audience can pick up on the multi-drug resistant issue, and if there are any specific programs throughout the state that are specifically screening for that in a large way. Does anybody have a comment to that specific question?
Brett, while we're waiting for that, I actually have something to offer. I think one of the concepts to consider is the traditional detection mechanisms that are performed to determine what the infectious agent is that's present that's causing the infection, and then frequently I'll use the term knee-jerk reaction to apply a specific antibiotic based on the way the physician has been trained or et cetera. The key, I think, in some of the innovation that's taking place right now is ensuring that at that detection method, not only that the pathogen is detected using a DNA extraction method and detecting the specific genetic sequence that's specific to that particular pathogen, but simultaneously testing to see whether they're drug resistant and therefore providing that information so that the problem won't get worse. I mean, it's difficult for me to really address the problem as it exists today, but I think you would agree it's important that we don't make it worse. And one of the things that is underway right now is ensuring that you can multiplex, that is, detect multiple organisms simultaneously and also detect whether they're drug resistant or not, and ensure that in that case the appropriate therapeutic intervention can be taken.
I will point you to Heather Manley, who's right in front of you, who is a program manager for a $1.7 billion program at the Defense Threat Reduction Agency, who is funding some of that work, and maybe you want to make the comment.
Yes, actually I do. So I just moved to Texas A&M University back in August, but before that, my previous life, as Brett just mentioned, I was at the Defense Threat Reduction Agency with a program called the Transformational Medical Technologies. That's a $1.5 billion program trying to address broad-spectrum antibiotics as well as broad-spectrum antivirals. And just to reiterate, this is a huge problem. By far, the weakest part of the portfolio was the broad-spectrum antibiotics issue, and multi-drug resistance, as you all know, is a major problem. Recently, a report came out of Congress that was looking at who was addressing this across the agency, and unfortunately, there are large federal resources that are available for this, but not enough research is going into it.
Okay, thank you.
And then just a plug for my former program, funding opportunities for those of you that are interested. If you go to www.tmest.org, Transformational Medical Technologies Initiative, is the tmti.cbdefense.org, or grab me afterwards.
Yeah, I'm sure no one's interested in funding opportunities, so, yes.
Thank you very much for 3 very interesting talks. I have a couple of quick questions for Dr. Girard. In terms of manufacturing, of course, the main issue is bioprocess development. And bioprocess development, as you know, is very modular, it's very system specific. Do you envision NCTM providing that capability? And if so, I mean, do you anticipate multiple platforms? And a related question is, as an end user, as somebody who develops biologics, how would I come to you, and why would I come to you? This is a little bit challenging of a question versus going to, say, Covance or some other private.
So let me answer the second question first, in that the NCTM is set up to be a core facility to enable the state of Texas academic faculty, students, graduate students, as well as biotech company to avoid the $5 million for your first gram of monoclonal antibody scenario that traps many investigators at the preclinical stage. So this really is a core resource that has been capitalized mostly by the state of Texas, and our mission is to serve the people in this room. If you're going to Covance and not going to us once we're up and running, we failed at our mission. So we want to do everything possible to enable products to get through. In terms of process development, yes, we clearly do process development both internally, and again, we have 11,000 students in the College of Engineering and departments who are working that because that's very important. I think one specific thing that's different is we are technology agnostic. We have another independent award that I did not talk about to create a vaccine rapid response center for the Federal Command Authority that says if we have a troubling sequence, particularly in biosecurity ranges, we want you to be able to develop the process. It might be in E. coli, it might be in CHO cells, it might be in EB66, it might be in insect cells, it might be in plants. Whatever it is, you develop the process with the right platform and then send it to the NCTM or the GreenVax project. So we definitely want to do that, and we want to collaborate with everybody throughout this room, throughout Texas. And we also have collaborations with the BTech Center developing in North Carolina for those same reasons. So let's talk again.
So this is terrific. That means that basically the cost basis for GMP, GLP talks eventually will be much lower through NCTM than through commercial entities.
Yeah, we're not
great news
implementing
for us,
until
yeah.
the fall, but we have a tiered strategy where Texas academia and biotech have a highly preferred cost basis versus some of the commercial customers that will keep us in business, if you will, to pay the bills. Yes. Of course.
Quick question for Brett and also for Patrick. Brett, this facility is very exciting and the technology is very exciting. It wasn't clear to me whether there was evidence that one could in fact produce influenza vaccine with the plant model. Is there a work that's shown that one could in fact produce the vaccine?
So the answers are in preclinical studies, both in mouse HAI and as well as about 30 strains of influenza in the ferret model, which is a feet up, feet down, best preclinical model. There's been high efficacy. There's been 2 phase 1 trials right now that are really phase 1Bs with 2 different plant-based products. One is a recombinant protein and one is a virus-like particle. Both show very high immunity and surrogates of protection, as you know, that would translate to human productivity. But I will say fully well that there's been no 300,000 to 1 million person phase 3 clinical trial that establishes that. This is a product development just like any other product development, but clearly the preclinical models and phase 1 studies look extremely good in terms of surrogates of immunity and protection.
Has H5N1 been, in the ferret model, been used in this regard?
Absolutely. In relatively good doses, these have been adjuvanted studies, but H5N1, H1N1, H3N2 new hot strains of H1N1 have all been in the feed up, feed down ferret model, and they've been positive.
Very good. Patrick, quick question. I was struck in my activities at the FDA that the personalized approach was very strongly resisted by the mature pharmaceutical industry. And the reasons for it was that they would much prefer to demonstrate 3 to 5% efficacy in 100,000 patients as opposed to 30% efficacy in 20% of those patients, given the market. Is that changing? Is it likely to change? Is it going to require regulatory effects in order to get a change? What are your thoughts about that aspect?
First of all, I would say that I would agree that you're correct, but I do think that situation is changing. One of the biggest factors that's changing that is actually the appointment of Francis Collins as the director of NIH, who of course also ran the Human Genome Project. He and Dr. Hamburg, who's the commissioner of FDA, have already done some very public things to try to fix that. Within industry, within big pharma in particular, for years there was a lot of resistance to the concept I refer to as personalized medicine. And the reason for that is because when you're selling 99% gross margin products, and whether somebody benefits from it or not, you still would like to sell those. Bluntly stated, it's a little more, a little more complex than that. But what is actually transitioned in the industry, and I think it will take some time to have this occur, is the transition away from the blockbuster model and to use some of the genetic information that I talked about that are generated during phase 1 and phase 2 trials to actually allow compounds that previously, using the blockbuster model, would not make it out of the pipeline, and have those actually make it into the marketplace, but use genetic and protein information as a way to segment patient populations and have products to sell in the future that otherwise would not have made it to the market. I have to acknowledge, I think that's a generational shift, though. I think it's probably maybe 6 or 7 years in the making, behind us, and I think we have another 10 plus years to go before it ends up being fully adopted. Within big pharma, the companies that are leading that particular effort are companies like Novartis and Abbott and some others, but I think it's going to take some time.
Okay, I think our time is up for the panel. Thank you, Patrick and Mauro, for a very exciting afternoon.